Hey, boys and girls. So we got an after show. How
I
you
like
feeling?
your logo. It was very cool logo.
Yeah. How you feeling after that?
Well, okay, pretty much I said the main stuff and I didn't say lots of other stuff which I wanted to say, but so
Yeah.
I mean the actual model was not presented in this discussion with Rebecca. But at that point I don't think it's so important because in fact if someone is really interested, there is a paper so you can go and just see the model. But I emphasize the several very important to me kind just for every creationist to know very well that they are tricking and they try to twist the actual model which the scientist is trying to work with. And by twisting the model, you present a wrong concept, which the scientist does not work with this concept at all. But you continue to push in this particular model. In the mind of Utangelo, for example, if he is listening right now, he thinks that all of the 100 prebiotic amino acids should smash together and make the genetic code immediately.
Yep.
That's not even the case. And there is no even scientist that thinks this way. It happens. So he creates a problem out of a wrong concept, completely incorrect and not viable way of thinking. So that's a kind of take home for Rebecca. I hope she did it.
Yeah. Yeah. When they ask for evidence, what evidence do you have in current empirical evidence? The point is that we have evidence for each of these small steps, for all the small steps, right? And she referred to a book, From Matter to Life, and a article by Sarah Amari Walker and Paul Davies called "The Hard Problem of Life," and she picked out a quote, where the one quote out of the book where they're talking about what a difficult problem this is, but this article is about how we need to that. One of the things that we're missing is more sophisticated mathematics dealing with dynamical systems. And then they talk about, the problem of not knowing the initial conditions, because we have a lot of unknowns there. And they also point out that there's no step in this thing that's impossible. And in order to prove ID, or in order in order to find evidence for ID, for intelligent design, you have to find something that is impossible. And there's nothing in any of these models that's impossible. They're just things that are problematic, the water paradox and all the other things that they've come up with. But there's no impossibility in any of these things. And nor are there many unknowns in what we know about the small steps, of the how one molecule reacts with another molecule, or how things behave when we raise the pH or lower the pH, that sort of thing. But yeah, and this article has a whole lot more into it. It's got a bunch of mathematics. I could put it on the screen here, because I do happen to have a copy of this book.
Yeah, I mean, Sarah Walker, I watched her. She's okay. I do not agree with her approach in terms of many ways. She
what
Sarah
is her?
Walker.
Go ahead.
With Sarah Walker, she's not defining the things exactly in terms of what you need for origin of life. And she's going in some kind of meta interactions. So it's a scientific approach. Of course, she's a scientist. She's a very good scientist. But her field is not origin of life. Even that she is writing for origin of life, she is not origin of life science.
Oh, sweetie.
What is
Yeah.
her field? Do you know?
Meta interactions of biosphere.
Okay, so that's why she.
Let's say it this way. I mean, I cannot say different. I mean, I don't know the actual terminology of Sarah Walker terminology because she's very prolific in terms of all of these interactions. So she has a team of cool programmers and bioinformaticians. She collects the data from entire biosphere for everything and trying to make meta interactions and those kinds of things.
Yeah,
But yeah,
she's working on dynamical systems basically, which is one of the things that we really need to.
yeah. I mean, dynamical system, but it's one to think dynamical systems which is already created and it's already proliferating.
Yeah,
It's different to look for dynamical system directly for origin of life, and she didn't do that.
yeah. She's talking about path, basically trajectories through what is it? The configuration space, that sort of thing. And so she's at a very mathematical level on this stuff, and
Yeah,
that's pretty cool stuff, though. And it's
she's
kind of what.
physicist by education. She's physicist. She's not a biologist.
So she's a
Uh,
she's working in biophysics or systems.
yeah,
Yeah.
it's not. It's not irrelevant. That's for sure. It is
Let's see if we
relevant.
can find the
But
conclusion. Yeah, they're talking about a new physics, basically. They shouldn't use words like that because people think they think of something that it's not. Basically, uh, dynamical systems as new physics. Dynamical systems are still deterministic in a sense, but they're but in order to understand the system, you have to look at multiple hierarchies, and you have to borrow a little bit from the future in your calculations, that sort of thing. But that isn't to say that the future determines the past. So yeah, I'm trying to find a conclusion for this paper because this is where she got the quote from. And the quote, what does it say? Why would you out of this entire beautiful book? Why would you just pick that quote? I don't. There are many
because
other
you did
quotes.
very well.
That's a good question for Rebecca. I invited her. I can't find my. Can't find it. The hell's my highlight? Let's see. Anyway, Scott and David, did you listen to the talk?
No, I was out this morning visiting some family.
Oh, that's right, you were.
Yes, I did. I listened to it.
Oh,
So what do you think?
Well, I think she's just, she's stuck on her view, I guess you might say, right? She's, very dedicated to her religion, and it's just a typical creationist things. I think, when she wants to search for any type of, information on these type of subjects, who does she go for? She goes for a Christian scientist that may discuss these topics that's not even in the field, like James Tour or Stadler, I think was one of them that she quoted.
Yeah.
The Paul Davies makes certain quotes like this. Paul Davies is an atheist, or agnostic, I think he might refer himself as, or whatever. But yeah, I mean, it's just typical. Do you call that cherry picking? Yeah. I mean, really, but that's just a typical thing. They're searching for anything that is going to help them out to strengthen their beliefs. Really, is what they're really looking for. They're not really looking for what's plausible or
For,
possible or any of that.
not for solutions.
Right.
They just looking for something to grasp
Yep.
and
Yep.
stay.
Yep. Yeah.
This is the quote that she read. The emergence of life in the mind from non-living chemical systems remains one of the great outstanding problems of science. Yep.
Yep, I agree.
Yep.
It is.
Yes, I vote yes too.
And whatever it turns out to be, it represents a transition from the mundane to the extraordinary. And life is the one example of an extraordinary complexifying system that we have on this planet. We need another planet.
And how you can translate that to impossible to happen.
Yeah, I know. Yeah. Yeah, and this article is making a case for this new mathematics or these new deterministic dynamical laws or whatever, talking about initial states and all of that. So anyway, that's on page 45. I'm trying to find the conclusion of this. What do we got here? This is a little illustration. Finite deterministic time reversible model. These reversible causal graphs demonstrate the concept that you can't always get there from here. Whoa. Composed of many disconnected regions.
I have a question that seems almost unrelated to this, but in a way kind of is. Last night I was watching a video about that 2 billion year old or maybe 1 billion year old natural nuclear reactor that formed at, and forgive me, I can't remember the name of the location. And it struck me as something that's very complicated, yet it still happened.
Oh, in South Africa? Was that?
Yeah, I think it was somewhere around O, begin with an O or something, the location. But yeah, it just struck me as something very rare, complicated, yet all the right conditions were there in order for it to happen and to last for about 1 billion years. Maybe a couple million. I don't know.
Yeah.
I'll have to go through it in more detail. But, the idea that, things are so complicated, so rare, so that it can't happen just seems to be disproven. Yeah, Okla, thank you, Nasli 20. It just seems to be disproven in nature all the time. I don't know.
Yeah.
I don't know what y'all might have to say about that, but I thought I'd kind of use that as an example of something complicated and rare, yet all the conditions came together and it happened.
Well, the interesting thing is that life is the one extraordinary system like this. It's the only system. Life is the only system like life, which so it's the only one we got. So until we find another planet with another system that perhaps differs, this is the only system we have to go by. And they're saying, "Oh, it couldn't have happened." Well, how do you know that? It's the only example we have. You see what I'm saying? We're kind of between a rock and a hard place when it comes to understanding life, right?
Right.
It's the only example we have. Let's see.
I mean, the idea why so life became proliferating different, very different, is because the actual selection is different. In many ways, you can select something in different ways. And if you pay attention about the information structure interaction and you have a feedback, that's exactly what the life is. If you have this system, you can have in a many ways, in so many different ways, all kinds of ways. And that's why it's so interesting because you have pretty much everything that you can think about. Yeah.
Yeah, it's incredible. And we kind of know it's incredible. I mean, when you go out and you look at the stars and then you ask yourself, how in the hell am I a kind of a thing in the world that can look at stars and think about them, right? And so yeah, we are extraordinary, in a lot of ways. Here's conclusions. Many difficult open problems in understanding the origin of life, such as the star paradox, Benner, which I think is a solution, not a problem. The fact that prebiotic chemistry is just plain hard to do. These problems differ qualitatively from the hard problem of life as identified here. Let's see. Well, ultimately reduced to known. Okay, while challenging, right? Okay, yeah, now, why not pick this quote? So most open problems associated with life's origin, such as these, while challenging right now, will likely ultimately reduce to known principles of physics and chemistry, and therefore constitute, by our definitions, easy problems. This is in the very same paper. Why not pick that quote?
Yeah.
So, yeah, we need to do more cherry picking ourselves. We need to have
we're too honest. That's why.
what we need is, when they pick a cherry, we need to pick an apple and throw it at their cherry. Just whack. And I'm glad Peter's not here, because
Yeah. I mean, they don't want these things to be solved, right? Because I'm sure it's for, you do hear a lot of Christians, they'll say, oh, I struggle with my faith. Well, because their faith is being challenged. So it's probably, it is a difficult thing to stay in the faith, and it's always being challenged. So a lot of these they just want to cherry pick to strengthen it, right? So that's just what they do. That's what they do.
The people that stay in the faith without denying science, how do they do it? They understand God as being so mysterious, so powerful, and so out there that they don't know what happened. So they don't have any literal beliefs about what happened, right? The only thing that they believe is modern science and the rational human beings who believe the science and yet hold on to their faith, because their faith is that, they have faith.
Right.
And,
right.
It's this deep spiritual connection that they have with what they call their creator. But they don't sit there and say, well, when did he create things? Was it on Tuesday?
Yeah.
Or, whatever. So, I mean, they don't concern themselves with those questions. Yeah. So I don't understand why the only way that a person is going to be challenged is if they are brought up in a kind of Christianity that is rigid and dogmatic to begin with. And then they start to find, the science is pushing back on that. I mean, if you believe this Bible is a science book, you're going to have a problem when you go to college.
Yeah.
Oh yeah. The kid is going to have a problem. So I mean, the solution
you go to a private Christian college that peddles to creationists.
Right. The solution is not to deny science. It's just not to stay away from literal interpretations and just get on with your faith, and live like a decent Christian instead of doing all this pseudoscience. There was a guy in chat. What did he say? Oh, okay, Angelo. He was saying that Dimiter. I don't know what he was saying about you, Dimiter, but it wasn't flattering. And I, and I said,
I'm not surprised, and I'm not arguing at the moment with the tangible. It's simply what is the situation with the tangible, and I will repeat myself because I'm presenting something which is the model how we are thinking about origin of life and the process of origin of life, the selection process, and he's still continuing coming and push his own model of smashing model. Of something super complicated that it's happened just by smashing all the elements in the science, and that's never happened and won't be happened this way. I will say, let's make one analogy between analogy of my discussion with Otangelo, and I hope everybody will understand this analogy. So when I said, okay, we're going to the moon, and Otangelo said, that's not possible. You cannot go to the moon. I said, why not? Because you cannot jump that high to the moon. That's not possible. And I said, okay, we're not going to jump to the moon. We're going to build a rocket, making a big, huge rocket, and blah, blah, mathematics, everything, technical stuff. And then we'll going to the moon, and then we'll be there. And he immediately will come back and say, well, I told you, you cannot jump to the moon. And I'm trying again to explain that we're not going to jump to the moon, we're going to build a rocket. No, you cannot jump to the moon, that's impossible, and continue forever. So what to say? I mean,
But-
I'm just stopped.
God can pick you up in his hand and set you on the moon. So Otangelo finds that to be the more plausible.
Oh yeah, that's another option, of course. Yeah.
Can pick you up and set you on the moon. That's more plausible to him, and he always says
So
that over
that's-
and over again. What is the most plausible? Or
So that's
there's
how
another
we're
guy that uses
discussing.
that
Yeah.
word. Yeah.
That's the way how we're discussing with the thing.
Irrefutable or refutable.
Refutable. What's up?
Refutable destiny.
Ah, man, I didn't watch the video, although I saw one of the comments saying like, kind of going after like, oh, Dimiter didn't address like Stephen Meyer. From what I understand, Stephen Meyer isn't educated in biology, so I'm curious why
he
he's.
isn't. Yeah,
I'm curious like why he's picking this fight to begin with.
he's one of those philosopher weenies.
Stephen Meyer, again, he presents a situation where it's not even correct. I mean, you cannot have, there is no way to begin the information to start up. There is abiotically. So you must have intelligent design to have information because information is known to be intelligent. That's his view. And this view is completely wrong for many reasons. So, but
Well, I mean, what's really curious to me is that like a lot of creationists, they're not paying attention to the modern literature surrounding abiogenesis. Like they're very much so dealing with like what we knew from abiogenesis in like 1960s, 1970s, about that time.
and also
during
what
the
is
time of
presented to them, what is presented to them to please them from Discovery Institute, because that's the goal of Discovery Institute, to misinform the people. That is the idea.
Yeah, and they're not actually interested in answering their own questions about the origin of life. I mean, geez, we saw James Tour earlier this year try and go after the origin of life, and he didn't even understand some of the most basic concepts that that kind of play into that field. And if anything, I would give him the benefit of the doubt and say that he probably does have enough of an understanding of chemistry to give like valid critiques on the field in regarding to certain techniques regarding synthesis or things of that nature.
Yeah, I agree. He is a chemist, and he's absolutely educated, well-educated chemist. He knows the chemistry for sure more than me, but that doesn't mean he is correct. The chemical part is one thing, the method and the actual way of thinking and the represented model, it's basically a totally different thing.
Well,
If
there's
you
a difference
continue-
between.
Yeah, for example, for James Story, we continue to insist that we have to make the car just because the car is sitting on the all of the parts of the car is sitting on the beach, and you must do it by itself. If you insist that model, obviously is wrong. So what to say about? Yeah.
Right, he's taking his general perspective, if I'm not mistaken, is that you have to have everything at once, all in the same place, or else it's just full stop. Nothing useful comes out of the situation if things aren't the way they are from the get-go.
Exactly. So if you put everything in the one tube or flask, so what will happen? I mean, you'll come to the best equilibrium at some after a few minutes, and that's it. We'll
Yep.
stop.
And I think there's an important point you touched upon as well, is that like there's a difference between somebody being educated in chemistry or somebody being educated in a particular field and being familiar like with the source literature that's available in that field. So chemists do all sorts of things. Some of them are concerned like with toxins produced by microorganisms. Some are some interested with synthetic biology. Some are interested in ecological processes or environmental processes dealing with geology. And each of those fields, like it has its own established literature for what people have done, what are acceptable methods, or what are methods that are conventional to that field. And the people in those fields are familiar with that literature and play by those rules. But for some reason, when James Tour is going after the origin of life, it doesn't seem as if though he's really taken very much time to read up on like what the history of the field is, or at least, within the past 20, 30 years, what methods are being used to kind of probe the study of the origin of life.
Well, one thing that I noticed, the James Tour things were 13 videos, and I must admit I only watched a few minutes of them. But one of the things that I zoomed in on was that he said that the cell membrane, okay, the outer lipids and the inner lipids are different, right? There's a differential in cell membranes, like phosphatidylinositol. I forget which is stronger, is distributed differently depending on the organelle and depending on which layer, whether it's the cytosolic layer or the extracellular layer, right? So he said that would be that would pose a impossibility for life to develop because there had to be different kinds of phospholipids in the beginning. Well, then in another session, he's talking about the tar problem, which basically, and the problem that there are many different kinds of phospholipids. So, but he's referring to this difference between the inner and outer membrane, right? And to my knowledge, that is going to be a much bigger problem in eukaryotes that have different organelles because of how the phospholipids end up in the membrane, right? But there's nothing that keeps a cell from forming with a pure one kind of phospholipid. It will form that lipid, those micelles, and it'll form the bilayers. And so that was one of his big points, and he was all cocky about it. And it's like, okay, Tour, you're wrong right there. Where else is he wrong? That's just one thing, right?
The two big things that are going to influence that, I would imagine, right? This is my best guess, is going to be, it's going to depend on how many different types of like lipids you have near, like in an area and in an environment that allows them to concentrate.
Well,
right.
They're created in the in endoplasmic reticulum and the Golgi, which puts on the inside of the Golgi, that ends up being the outer face of the cell because it flips, right? Flips over. So whatever's created on the inside of that is where your concentration is going to be. But then if you don't have Golgi apparatus and you don't have an endoplasmic reticulum, there's going to... If prokaryotes have this difference, and the difference is about whether or not you have negative or positive charges on the inside and the outside. So, but those things are not necessary to the protocell. The protocell only has to encapsulate a chemical environment. Okay.
Oh, you have to have some sort of compartmentalization. It doesn't matter. Even you don't need to have exactly lipid compartmentalization.
Yeah.
Of course, it's most likely to be lipid, but in theory, that's not the major point.
Yeah. So I mean, his criticism was just, he was so sure of himself, and he was just way off on that because it's a much more complicated situation than he was portraying it as. Oh, this is impossible because we couldn't end up with this differential between the two membranes or sides of the membrane. And that irritated me. And I'm guessing...
Well, he also has a different argument with that, too, is just like the van der Waals interaction between lipids has to be a certain way. And it's just like, wow, really?
What was that about?
van der Waals interactions are interactions that occur between kind of like different parts of a molecule, and they allow molecules to stabilize.
What was it?
His point was just like there's this very fine like chemical detail that stabilizes, that allows the stability for lipids, and if it wasn't, he's trying to argue, if it wasn't exactly the way it is, it would be impossible.
Yeah, but it is that way, and those things form really easily. I mean, it's a real thermodynamically favorable state for a lipid bilayer to form in water. I mean,
right, but he's going to argue just from pure probability, right? He's going to take arguments that ignore like why it is that van der Waals forces between lipids are the way they are.
But it's highly probable. You know.
I mean, to tell you experimentally, I think Bruce, he actually did experiments with the different lipids in the different conditions. He put with the salt conditions, different ionic conditions, with different salts. Yeah, Bruce.
Yeah.
And he published this data, and not just salt condition and pH conditions, and he tried to sense and to see which lipids actually might do it. That's very feasible, very reliable experiment, which we know what happens with lipids. It's very
Remind
difficult.
me if I'm to add to your point. I believe Shostak, I think this was a while ago. It might have been like 2008, 2010. He did a few experiments on this type of thing where, if you imagine protocells having different lipid constituents for their membranes, right? And he was trying to address the situation of if these systems are in a state where they're allowed to replicate, right? What type of system? What type of equilibrium does a system achieve, right? Do you get systems that have, for instance, do you end up with systems that have just one type of lipid? Do you have ones that are that have multiple kind of lipids? Do you have a preponderance, or is there any selective pressure for why phospholipids get incorporated into that type of structure? Things of that nature. So I believe Shostak a while ago has done some research on that.
I'm not familiar with the super greater details, but I remember he did lots of experiments with the lipids actually. And actually, in these conditions, the lipids are not like a vesicles like this. In these conditions, the lipids are like a macaroni. They're kind of more elongated type of lipids. And also, what he showed that it just naturally they actually divide as a lipid. So, and that was his. Yeah, you're right. Before 10, 15 years ago, that was kind of his major topic of research. And of course, his idea was to put RNA inside. So the whole idea is to put RNA inside the bubbles and to show that the RNA can do whatever ribozymes can do based on in the lipid environment. And
this is,
of course,
yeah. This one is just by Dimiter on lipids. I'm not sure. Is this?
Yeah, probably that was the paper. It kind of relatively recent paper. Yeah, 2017. Yeah. So, and he did a good research actually for this particular problem. And nobody denies. I mean, you have to have some sort of compartmentalization. There is no other way, and nobody denies that. You cannot have those reactions of interactions between RNA, peptides, even lipids, just in the middle of the ocean. I mean, not going to happen, obviously. And actually, I remember he actually picked up actual water from different sources. Yellowstone, Kamchatka, maybe he picked up.
Yeah.
Oh, most likely no, actually not. he actually measured the concentrations, ionic concentrations and ionic conditions of different water, and he created it back in the lab just by putting all kinds of ions like magnesium chlorides, blah, blah, all of that stuff. And he have a good recipe for what is the ocean water, what is the spring water, what is the water in the uh, geysers in the Yellowstone Park. That's a good research.
Yeah. Interesting.
But so my point for origin of life, they all of these conditions matter. All of this research is important. The prebiotic research is important to show how the nucleotides are formed, how different amino acids are formed. I mean, those kinds of things are extremely important, and we should be very thankful to the chemists that are doing this. John Sutherland shows that cyanide chemistry pretty much can go to almost everything that you want in terms of prebiotic conditions. If you just UV, I think was for carbonate, some carbonate was, if you just UV irradiate this carbonate, but was more specific carbonate, I don't remember exactly. You can have all kinds of species. Pretty much half of the Krebs cycles might appear directly from this particular experiment. So it's no longer kind of mystery, the prebiotic condition. Yes, it's not known. We agree completely. We don't know exactly the conditions, and it's a problem. But that doesn't mean it's not solvable. We see it's probable. It's highly likely to happen.
Well, I mean, this is kind of cheating, but part of it is we know it's probable because we're here, right? Like it seems that if we're impossible, the type of chemistry and the type of system like that we're using, which is far, far more evolved than like what it started off as, wouldn't be able to form in the first place.
Yes, right? And also from if you read ever, I mean, this is my third paper for Origin of Life, the point of no return. You will see there is a section about the probability of life, and yeah, the argument was very clear. The fact that you have so early fossil evidence, and showing very, very early kind of origin of life, pretty much in the first 200 million years after when the Earth chilled down, that by itself want to say that the life is very probable. You can think you can think differently. Let me say, all of the fossil evidence shows that the very first fossils appear about 1 billion years after the Earth chilled down. Let me say it this way. That's not the fact. This is not the fact. But I'm saying you can think that way. Or let me say 2 billion years after after the Earth formation. So if you have the first evidence for life after 2 billion years, that's a different story. Now you can say, okay, now probably the origin of life is something very rare. It's something very unlikely, and just it happened in the planet Earth somehow very late after the first formation. But that was not the case. We know very well that the origin of life happened extremely early on the planet Earth.
So, yeah, to respond to Nestlig, yeah, the chemistry is kind of unusual because it's using free radical chemistry. But the reason he's pursuing it is because the types of substrates that the synthesis is drawing upon are very, very ubiquitous and very, very strongly like present on the early Earth. And so even if it is chemistry that's not typical for life to use, it doesn't necessarily need to be.
Yeah. So
But the other thing as well, to add to your point a moment ago, so what's kind of curious about the origin of life, at least as we observe it, like with our planet, is that about 500 million to a billion years ago, or after the planet formed, prokaryotes formed. But only a billion or so years ago did eukaryotes form. So for some reason, like there's an extremely long, well, if you talk about the Earth's lifespan, an extremely long span, extremely long gap in time between the development of eukaryotes from the development of prokaryotes.
It is absolutely accurate. The reason for this is event, a single event which happened at that point. So to have
the great oxidation event.
Yeah, not just that, but the mutual benefit, for example, let me say between the bacteria, which it's happened to be the future mitochondria, that's a long time ago, or the event of multicellular organism, which is happened even much later. It is all about unique event, which is actually happened because of the mutual interaction between the organisms. Now you have an ecosystem. So imagine you have ecosystem, and now, of course, we have a mutual beneficial organism. So we know very well every organism, pretty much there is no organism by itself, right? You have organism which exist with a huge biome inside. There is no organism without biome or interactions with the other organism. And at some point, evolutionary, the benefit between these two, it's just happened. And voila, you have another organism which is merging from two different organisms. Now we have mitochondria and chloroplasts.
Right.
It is all about, and also the people are very confused. They think that the evolution is something that should have a goal to make a something, like have a goal to make a human. No, that's not true. We humans, the high level of organisms, it's appear just because the Earth slowly and gradually changes the environment, changes the conditions, and you have another selection to come. That's why you must understand, and everybody should understand, that the evolution does not have a specific goal. And the only goal is surviving. That's why there is living fossils. Living fossils exactly that proof. You have organisms which are living in non-changeable environment, and they stay as it is.
Yep.
That's the question. The question of transitional species, for example. Imagine hippopotamus, which usually the little girls likes for Christmas, right? Hippopotamus, which is the best gift for Christmas of some little girls. It's a transitional species. It's a transitional species. So they're living mostly in water, but it's obviously land animals which are walking around. Now, in what conditions this hippopotamus will become like a manatee? In what conditions? I mean, it's obviously once the land becomes more wet and wet. So this is the moving of the continents. And one became more wet. Suddenly there will be some species which will be more adapted to more longer period of time to stay in the water. And you need to have a changing of the conditions. That's why hippopotamus can turn in the future into something like manatee.
Well, it wasn't a hippopotamus. It was like a common ancestor of the hippopotamus and a manatee.
Well, the common ancestor is the common ancestor of the elephants and manatee. So a long time ago, there was that process of going, the landscape became more wet, wet, wet. Suddenly, it is more prone to survival in this way. So let's imagine you see that manatee, for example, it's eating grass, it's eating algae, right? So if you're specialized to looking mostly for this, and as many and as longer and longer you're living in this type of environment, of course, suddenly you'll be more adaptable and you'll no longer need surface. I mean, the same process happened with wells, the same process. And also you can have examples on the modern transitional species from sea to the to the to the seaside, to the land, right? Mudskipper, for example. If you see mudskipper, I mean, what is the best evidence of transitional species? Just show mudskipper. It's exactly fish.
Yeah, I've seen videos of them. They're quite remarkable. I mean, they live in water, but they spend quite a large. I think their breeding cycle actually, like for them to breed successfully, they actually spend quite a lot of time on land, though.
Exactly.
Kind of like transitioning between the two.
Now imagine a situation where the land became more vast, and just you have from time to time wet cycles and so on, just to keep them breeding and so on. And suddenly you'll have a situation where the mudskipper will no longer need exactly full time to be in the water. That's how, I mean, the people don't understand that the evolution, it's not goal to make a high level of species, more advanced species or more complicated system. It's only about surviving. That is all about. Parasites, it's another example. Parasites, actually, they became less complicated in the evolution than they was initially complicated. Now they became less complicated parasites, right? Flatworms and so on. Hey, Shadow, show back this mudskipper. It was very cool, yeah.
The real question is, why did birds evolve? Because apparently your bird is very active. It's very active today.
They was chasing dinosaurs.
Right?
Don't want to upset you there, Dimiter, but there's a vulture on you.
It's a very adorable dinosaur.
It's a big vulture.
And the birds are very curious. Here we go. That's the mudskipper, yeah. I mean, what is better than this as a transitional species? Come on. If someone tell me and have a doubt about transitional species that we find in the fossils, I mean, what about the modern transitional species? Look at that.
Yeah. How much more do you? What more do you want?
What more do you want? Exactly. Yeah.
We have these fish in Minnesota that you catch them and throw them up on the shore, and they start to wriggle and crawl right back in the water, and they can live for like 24 hours on the shore. So,
wow,
what is that, lungfish, I think, or something, or we call them dogfish. I don't know if that's what they are. That's not really what they are, but anyway, they're
is my mic working?
Yeah, it is.
Yeah,
thank you.
it is. Yep.
Yep.
Yeah, I do agree with your last comment, Nestle, regarding Sutherland's work, and that is, it's important to note like that UV light and cyanide are both very dangerous, like to modern organisms. So even if Sutherland is correct in terms of it being able to form these processes, being able to form nucleotides and lipids and what have you, it's a little bit unusual, because if that's the case, why doesn't life use cyanide or, rely more heavily on UV light than what it does? Because if that were true, if that was a potential source for early metabolism, then cyanide would be something useful to the body rather than something that kills it.
Well,
Yeah, but why would that?
The answer. Yeah. Okay. So.
To be fair, it is from Nick Lane, not my specific.
No.
Yeah. In any case.
The answer is because so the origin of life, when you have Terra Darwinia is dependent 100% from environment. and from the sources of environment. And what is happening that during the evolution of Terra Darwinia toward the Luca, you have a process of independence from the environment. So if something is very rare, for example, arginine as a amino acid, although it's probably very important at the beginning for interaction between RNA and peptide, it's rare. It's not
Yeah.
much there. And what does it mean? That means the first complexes will not go too much further initially, but they will still go somehow in next evolutionary step. Next evolutionary step is just recruiting another amino acid to the peptide. That is the next evolutionary step, and that might help for something else. But yeah, okay.
Yeah, and there's another point that's really related to this, and that is this critique of like quote unquote ancient chemistry that like the prebiotic systems might have used. Well, even if it's not traditional or something that's very unlikely to have occurred, right? Remember, these cells are very basic, and they're using whatever possible ways that can generate these resources that's available to them. Now, once you actually have like the formation of the genetic code occur, and you have things where cells can actually begin to reliably synthesize, amino acids and nucleotides and things of that nature, then the chemistry will probably change because there's, restrictions on what on the type of chemistry proteins can do, and what chemistry is destructive to those things, and so on and so forth. So
yeah, exactly, and then the life can move on, basically.
Right. So it makes sense that there would be a phase transition of sorts where, the chemistry gets kind of scaled down to a form that life can use and that's not harmful to it.
So that's why that's what you said. The Terra Darwinia is 100% heterotrophic organism, so there is no autotrophy there. Heterotrophic meaning it entirely depends from the environmental sources. And once you learn evolutionary how to make something else, let me say arginine, now you can skip and keep going much further and much faster. You'll be one step independence. And so on and so on. My view, which I expressed in this paper, the last paper, is that metabolism is a way of independence. That's what is the metabolism. Way of independence from the primordial conditions.
Yeah, I think I would sympathize with that sentiment.
Yeah. Any other issues, guys, you want to discuss?
Issues.
I
got issues.
I just came in. Like I didn't know that you had the conversation with somebody else before this.
Yeah, with Bread of Life.
With Rebecca, Bread of Life.
It's linked in the description, the original
right.
conversation. So I hope.
It was also on the origin of life, right? Probably.
Yeah.
All right.
Yep.
So we need the good experiments, unfortunately. And also I have to tell something, the scientists are they're normal human beings. They don't read everything which is available. Unfortunately, I don't know everything which is right now on the literature. And the other scientists also, there are lots of scientists that haven't never read whatever I did, whatever I wrote down.
And to confound the problem even more, right? Relatively speaking, the field of abiogenesis is becoming more active, and the amount of data becoming available on it is expanding rather fairly quickly.
Yeah.
So, it's not as if though you can just read a paper a day and be fairly caught up with it. These papers are,
But the precise moment, so what I want you guys to take home as a basic lesson is that in order to have distinction between chemistry and life, you need to have Darwinian evolution. That is the distinctive point. Also, we can name it informational dependent selection. Information dependent selection, which is the same, basically, Darwinian evolution. If you don't have that particular moment, there is nothing you can advance. There is no way to advance. You may have initially assembled a huge, enormous polypeptide or something by chance, or some kind of polygoo of huge, complicated stuff. But those things without information dependent selection will stay at the same level all the time. And if it's formed, will form again in the same complexity, in the same, it will never advance. That's why I want to emphasize very clearly. Once you show me how the information dependent selection occur, you're showing me origin of life.
Yeah.
That's it.
Yep.
I did have one commentary, although maybe I don't know if you want to continue with this, or anyone want to continue. Like I've made a comment.
I don't mind.
I've made a comment in private chat. Like I'm more familiar about the metabolism first view, and they say that metabolism is more of a way to resolve the disequilibrium between, or the redox disequilibrium between hydrogen and carbon dioxide, instead of like being independent from your environment.
I tend to fall in love with what I know the most about.
Well, the metabolism first. Unfortunately, it's not one thing. Some people consider metabolism first Tayo worlds, having nitro worlds or something. Another people consider metabolism first the gradients, which is happening in a deep black smokers, deep vents. In every case, you have some sort of chemistry, and nobody doubt that you have chemistry. But yes, you may describe lots of goodies happen in the boundary and the sources of interactions. However, what you can the definition for metabolism, to me, it's something which is organized from already existing life. If you have metabolism outside of information, you pretty much are just having some kind of likely reactions which resemble whatever is happening after life, after the life is formed. So I agree to some point that those kinds of research about the oxidation level, for example, oxidation levels, and you must have a synthesis between carbon 1 and carbon 2 and carbon 90, and those are important, but those are not necessary to happen in a way that it's specifically informative to make the information, to make a selection after that. So that's my point. For example, the autocatalytic reactions, there is autocatalytic reaction shown experimentally. So you see it as a cycle. Actually, it's happened this one, turns in the other one, and
Right.
after this one. And this is autocatalytic reactions. Now, okay, that might be important, but show me how this will turn in an information-dependent selection. Again, back to the point I'm trying to say. See, this is the master lock of origin of life. Master lock. It doesn't matter from what kind of hypothesis you will come. you must show me how information-dependent selections come to be. If you don't show me, well, could be important, but it's not origin of life. That's what I'm trying to say. And the people don't understand and don't understand, continue not to understand. Even the scientists simply trying to wiggle, trying to show how information will appear first, but the structure will appear later. No, you cannot do that. Simply, there is no law. There is no possibility to have information outside of what this information is doing. You understand? So that's why I'm not a big fan of metabolism first, from the point of view that the metabolism first you can go with life. You still can have metabolism first reactions, papers, goodies, important stuff, but there is no way to call this life. That's my view.
But it can be a precursor to the information-dependent
Of
selection,
course,
right?
yes,
Yeah.
yes, I agree with that, and I wrote it down. Yes, you can still have it a good prebiotic chemistry and precursors based on metabolism-like reactions. I agree with that. There's no problem with this. The problem is that you constantly call this life, and that's a huge problem because you don't have upgrade just based on this.
Right. Like I am not familiar like how they would explain like the origin of like the relationship with immutable is also going by immutable, but I have seen that they like one of the curious thing about for example about the genetic code is how the first letter of the codons relate to the like even the first letter of the codon to the amino acids. Like the first letter is correlated with the metabolic precursor of the amino acids, and then the metabolism first hypothesis guys want to make a connection from metabolism to that. Although I don't know how they specifically go about doing that, but it sounds pretty interesting in the connection from first metabolism to the genetic system.
Okay. So let me explain to you what they mean. So it's a so-called there was a specific term for this hypothesis for origin of life. And actually, it's not just for origin of life, it's about the evolution of the genetic code. So the idea goes like this. The very first amino acids are the most simple ones. And the most simple ones are more close to the Krebs cycle. If it's closer to the Krebs cycle, that means those amino acids are the first integrated into the genetic code, assigned for a genetic code. And that's the logic about this hypothesis for evolution of the genetic code. Now, I have a huge problem exactly with this particular model. Although it sounds very convincing, it is true that the most simple amino acids like glycine, alanine, valine, isoleucine, those amino acids are kind of very abundant in all of the prebiotic simulation experiments that has been done, and there are more than hundreds of those, like a Miller type of experiments. So in different conditions, how and what you can have. The fact that some amino acids are more abundant than the other does not make automatically more valuable for the very first compounds. One amino acid could be extremely high abundant, like glycine, for example, and at the same time to do nothing for the very first RNA peptide interactions, or to do very little. Now, The idea that once you have a very simple amino acid, those are linked with the Krebs cycles of the first that are coded, and after that, when the actual metabolism start to grow, next generation of amino acids will be assigned in the genetic code. That idea could be correct only for the latest amino acids. For the amino acids like the last five, 6 amino acids. But this concept cannot be correct at the very first one, because at the beginning you need to have specific function. That's why in the paper which I dedicated for the origin of and the evolution of the genetic code, which is beautifully, very well fit in the RNA peptide flow, that's exactly what I stressed out. You cannot just go and say, okay, that was the first amino acid, that was the second amino acid. No, you must figure it out. What is the function of this particular amino acid for the survival of the first life? And that's what is important. You cannot just go and try to push based on some logic ideas. And frankly, to tell you, advancing under the biotic conditions metabolism to go, you need enzymes. Although could be a very primitive enzymes, you still need amino acids to be incorporated. So the actual organization of the metabolism by enzymes require a very well developed genetic code. So it's kind of catch-22 situation here.
This is actually, oh yeah.
Yeah.
Like if you, I want you to finish first if you are finished.
Therefore, I want to say the most important thing you need to look when you're going to the evolution of genetic code is what actually any particular amino acid contribute to this particular level of evolutionary step. That is very important. And of course, that's kindly very unknown, but I found one thing which is very important. And this is the evolution of the peptides and making from the peptide to the protein. So as I said, the difference between peptide and proteins are very clear. Peptide interacts and do whatever it's doing just based on the amino acid sequence that it's there. So based on the property of every individual amino acid that is in the peptide, you can do stuff, you can do things. However, once this peptide is very long and make a fold, 3D fold and structure, now you have catalytic centers. And now you have domains, you have motifs, domains, and so on. So the evolution of the proteins goes from peptide, going through motifs, domains, and finally you have a fully developed proteins. That's how it works. Well, in that case, the very first amino acid that are incorporated should be very beneficial for short peptides. The next generation of amino acids which are recruited to the genetic code should be very beneficial for these folding structures, 2D, 3D structures, and making catalytic centers. And the final levels of these amino acids should be important for the beginning and the end of the proteins. So, and that's how the logic goes. And if you see this, if you try to figure it out, all of these functions, and you can read in the paper very well is described, you will see that most of the first amino acids, which is the arginine, proline, glycine, and alanine, actually have a very good sense to be a part of the peptide which interacts with the RNA. Not only, of course, not only, there is many more things. And the next generation, and that happened to be a GC-rich coded amino acids. And the next is a mixed generation, And finally, of course, you have amino acids, which is about the beginning and the end, and all of the stop codons are there. So you must find some sort of logic, functional logic, not just mathematical logic, because you may have a very good mathematical tables of codons, but you must have a function. That's what is important. That's what it is. So I don't know, quite clear or not.
Right. Well, there was one thing you said about like how you need proteins for metabolism.
Yeah.
Yeah. Although of course I'm not of course an expert on this, but I've heard the I've read some of the book by Eric Smith and Morowitz, and they point out that the enzymes who perform catalysts, they often rely on like cofactors for their catalytic activity.
Yeah.
And in core biochemistry, where carbon fixation happens, these catalysts are metal centers. They are minerals, so they point out like, oh, it's almost like mineral chemistry is enfolded into proteins, which came later.
Now, from one side, that it's not controversial, it's not a problematic for RNA peptide work. I mean, you have initial complex which can rely on whatever is happened around. And whatever is happening around could be, of course, some sort of reactions which resemble and produce building blocks. And that's not a problem to be metals, metal ions, all kinds of surface-driven reactions, different types of energy source like a UV or heat or whatever you can say. And that's, or even boundary. But what I'm trying to say here, it's not that you cannot have initial reactions which resemble the metabolism. My problem is that those reactions must be organized from something which is already informational dependent. In order to have the same effect after that, you have to have the same outcome or even greater outcome next time. You must have information presenting somewhere. You may have a perfect metabolism everywhere and there is no life.
Right.
Why it's not life? Because cannot evolve. I mean, in order to evolve, you need to have RNA peptide work.
Right. It's like I don't think there's a disagreement with what you were saying. Like, the metabolism they are also, of course, think that the DNA and the RNA relationship is important for the origin of life, and I think that's also a good thing to investigate, like how the genetic system between the relationship between the RNA and the amino acids happened. And this is something I'm also looking at. Like you mentioned a paper, let's see, the evolution of the genetic code by Lee and Burton, right? Is it the paper that you were talking about?
Yeah, Zachary Burton.
Yes.
Well,
I've seen that paper too. This is a very good paper, yes.
Yeah, so what I'm trying to say, we discussed with him, we shared several emails together. And of course, as a scientist, I agree with some stuff that he provided and hugely disagree with other stuff that he provided. And for example, what I agree with him is so-called microhelix world. So the microhelix world resemble these short RNAs, which is handled around and have loops and hairpins and so on, from which actually arise the tRNA. and the other RNAs, like proto small subunits RNA of ribosomal or proto large units of those kinds of things. And this is perfectly well fit with RNA peptide world because that stuff is exactly what you should expect in the evolution of RNA peptide world. And he provided very good model, for example, how the tRNA actually evolved out of first 31 nucleotides, which is just flipped and copied and just amplified and make a tRNA.
The first 21 nucleotides, are they the place where it attaches to the amino acids? The attachment site to the amino acid?
So he's looking, yeah, but this is about the evolution of the actual tRNA.
tRNA, yes.
Yeah, the actual tRNA, which I'm perfectly fine. I mean, it's perfectly correlate with whatever model I present. It's simply in more details. So
according to him, the anticodon came later. First, it was the attachment site to the amino acid, and then the anticodon part of the tRNA.
In his view, so he has his model. It goes like this. Initially, you have RNA world, and this RNA world somehow is capable to produce polyglycine and by ribozymes. But the RNA world means you also have RNA, a ribozyme which do self-replication. That's what it is. But in this RNA entity, one of the ribozymes, it starts to play the role as amino acylation and make a polyglycine. And this polyglycine, according to his model, actually serves as a very good feedback to stabilize the proto cell by making bridges around the cells with the polyglycine. And actually, any system that produces polyglycine, actually it's stabilizing itself because it's not in direct way as I presented in my hypothesis. In his hypothesis is indirect stabilization. So whoever it's making polyglycine, it's actually more stable according to this model.
So it's basically the first step in information-dependent selection. The information is the making of glycine, and that helps survival of the whole system.
Yes, according to his model, is that. Again, according to his model, later on, first amino acyl synthetases, which is a protein, it just suddenly appeared and started to amino acylate glycine only. So the very first amino acyl synthetase was the glycine amino acyl synthetase, and of course the proto-tRNA is the glycine proto-tRNA, which started to have codon, which actually involved all of the nucleotides, but producing only glycine. And from that point, you start to diverse. So the first nucleotide of the codon recruits one glycine. Once you add another nucleotide from the codon, now you diverse in the more amino acids. They became 4, 8, 16, and then you have pretty much, once you are 16, you have a transitional point where EFTU, which is the ribosomal factor, which increased dramatically the recognition of the third base of the codon anticodon. And actually, that moment, it starts to recruit all of the rest of the amino acids.
Right, because in the past, the first and the third codon were wobble base pairs, and they weren't reliable to be used yet.
And it was not recognizable well. That's why EFTU, once it's involved.
Yeah, it's basically the addition of that factor is basically to increase fidelity in the information system.
Absolutely, yeah.
Right.
However, However, I have a huge disagreement with this model. Huge.
Who's that you're talking about? I wasn't paying attention. I didn't get it. The polyglycine guy. What was his name?
Zachary Burton.
Burton?
I put a link in the paper in the private chat.
So,
He's one of the authors. Yeah.
Zachary Burton published his model maybe around 10 times so far in different years. The same model, he published it many times.
Okay.
In different ways. So whatever he presented was presented in back 2016, 2018, actually 2019 probably was the first papers and so on. Anyway, so I have a huge disagreement with this model. First disagreement is the RNA world by itself. So you have a system which produces polyglycine from RNA world, which, as I mentioned, it's very unlikely to have RNA entity only because of this requirement for the self-replication ribozyme. There's no such animal self-replication of the RNA ever. So I completely disagree with that moment. The second moment about the polyglycine. There is no answer to the question, why should be polyglycine? There's no any reason to be a polyglycine. In fact, polyglycine is one of the meaningless amino acid in this particular fashion. Yes, it could make some kind of net of polyglycine to kind of hold the protocell together, but in fact, that could be done with any other amino acids, also very abundant. Even there might be a valine or isoleucine, which might do pretty much even better the same thing because you have also lipid layers around. There's no any reason to be polyglycine, and that's a huge problem. Why should be polyglycine? From his perspective, polyglycine must be because according to his model, the glycine is the first amino acid synthetase for the recognition of the glycine, and for the tRNA is the glycine tRNA. But simply you have a huge flaw here.
So instead of polyglycine, you probably think it's more likely that the initial system basically randomly polymerized the available amino acids, including other than glycine.
Yeah, I mean, you have around many other amino acids. So he did simply resample from the modern bacteria's walls, which using some sort of exactly this this is not coded amino acid peptides. So this is a peptides which have even not just left but also right amino acids. Which is incorporated and build up the wall of the bacteria. And he said, okay, because the wall of the bacteria contains glycine and contains other amino acids, in the same fashion in the past, he didn't say it's a direct descendants, but he's just given it as an example to say that, okay, you might have a good security by having polypeptide in this way. I didn't buy that because in fact the actual walls, the bacterial walls, does not contain only glycine. It contains many other amino acids, and also some of the walls of the bacteria even don't have glycine. So it doesn't make it special amino acids for this particular function. And that's one thing. Of course, experimentally could be shown somehow. I don't know. I mean, it's still open question. But my view is there is nothing special about glycine to do this.
All right.
And the second, several kind of completely unlikely events after this point. How EFTU will appear at some point as a fully developed, or let me say not fully, but almost fully developed protein without having so many codons before that.
EFTU is a protein, right? So yeah, it's
It is protein. It is protein which is doing very cool stuff. Actually, it's a catch the tRNA from one side, the opposite side where the anticodon is, and push it inside the A site of the ribosome, and also interacts with one very interesting loop on the large subunits. And now, if they fit very well, it's kind of happened like a turn, very interesting turn. And GTP, you have a degradation of GTP, and this particular EFTU immediately push the tRNA inside the ribosome if it fit very well. If it doesn't fit, it not goes. It's a very cool actual system, but dramatically increase the fidelity of the translation process. So if you look my hypothesis, and if you look the chart,
is it maybe?
Yeah.
Sorry. My question is it maybe possible for EFTU to have evolved before you increase the fidelity of this?
No, it doesn't make sense, because you have to have different function in that case. The only way, the function that, of course, the EFTU as a protein actually resembles another proteins. It's not like a unique, as any protein. EFTU is a protein which also resembles some of the amino acid synthetases in some stuff. So it's not like a coming from nowhere.
That's interesting.
It is evolutionary developed protein, which it's happened to be extremely valuable for increasing fidelity of the translation. However,
EFTU is a GTPase, right?
Yes, it is.
That's one of the oldest protein classes I think there is, right? GTPases. Right.
Sure.
Yeah.
Now, if you look my chart for the amino acid synthetases and the actually evolutionary development of the different amino acid synthetases and the steps in which different amino acids are recruited, you will see that I have two major steps. One is called pre-Luca 1, and the other is called pre-Luca 2. This is in my second paper. And I don't want to bother at the moment just to show you the actual picture. You can go and see it. But my point is, there is a very good sense and common sense how the things happen, because in fact, according to RNA peptide world, and particularly this hypothesis which I'm presenting, the initial landscape, the landscape of the RNA was mostly GC rich. You don't have equal representation of all of the nucleotide, and that's why it's happened selection just in a one way, the GC rich amino acids. However, at the moment, once you have a process which facilitate the RNA replication, now you have a conditions when you have incorporation of AU, more AU nucleotides. However, neither the first time, neither the second time you have only, this one, exactly, So neither the first time, neither the second time you have only GC or only AU. I'm talking predominant AU. And once you have a pre-Luca 2, you have a condition now where you have evolution of EFTU to appear. Because at this first pre-Luca 1, you already have a formed ribosomal structure, although this ribosomal structure is not developed too much. It's just you have a proto subunit 1 and proto subunits small and proto subunits large, which is doing translation, although this translation is very wiggling, very vague, it's not too certain, but still is working in a way to select glycine, alanine, proline, threonine, and serine. It's good enough because those are GC rich, right? So it's good enough to do it. And that's what is happening. So it makes perfect sense, everything. I mean, I don't see anything that doesn't make sense in this particular hypothesis. So that's why with Zachary Burton, although I respect him very much and he's doing interesting stuff, I disagree with many of his claims, uh, and he disagree with many of my claims. So he do not agree that the codon formation, or I should say protocodon formation, is the first thing that appeared. No, he disagree with that. I agree, he disagree. So that's the science how it works. Nothing, nothing
And,
usual.
if I'm not mistaken, he also, or at least according to his paper, the class 2 amino acetyl synthetase is the first family of
Yeah, I
But,
mean,
but,
I
but, in,
used
in, but
this.
in your image, it shows that these two have this like almost simultaneous origins, or maybe I'm misunderstanding the
No,
thing.
no. You can see very clearly in this picture that class 2 are the first.
Oh, all right.
You have so it makes perfect sense. Proto aminoacyl synthetases class 2 that are able to recognize glycine, alanine, proline are the first. Whatever you see is a green here is a proto class 1. This is a very simple, very close to the bridge peptide, which simply recognize one thing more. This is the arginine, nothing else. But this recognition is based mostly on electrostatic interactions. So not based on a shape of the protein, based on the shape, which is about the class. And in fact, the class 1 is developed in a later stage, in a pre-Luca 2. So it makes perfect sense. And also to make this particular figure, I used exactly the Zachary Burton's phylogenetic analysis of how likely and close are those amino acid synthetases, and to tell you perfectly fit everything. So I have perfect fit. So it's how it's possible to happen everything experimentally that it's coming from from the Zachary Burton to fit extremely well in my hypothesis, and that's it. So I use it because, and I acknowledge that his research.
And are you maybe also do you also perhaps suspect a explanation for what drives this whole system to acquire more amino acids into the coding system?
Now, to tell you the truth, the way from pterodarwinia toward going Luca, it's a very, very dark area in the science. It's very difficult to figure it out what kind of processes actually it's happening and what is the reason, for example, to be incorporated serine, for example. There is a protease, it's called serine proteases, and maybe that was the reason, but who knows? There is additional very interesting motifs which Lupus described. It's called KH motifs, which is strangely assembled only from class 1 captured amino acids like isoleucine, leucine, and valine. And those, very strangely, are interacted with RNA interaction. But it's happened in a pre-Luca 2, not in the very first stage. You don't know basically everything. You don't know why this particular amino acid is incorporated, and we don't know. I mean, there is a huge, deep, how to say, open field for research. Between pterodarwinia and Luca, It's like a terra incognita. Nobody knows. And this is a future research for maybe years and years of scientists will research that.
But perhaps maybe it's only part of the answer, but perhaps there's also a ratchet effect where once an amino acid gets incorporated, it becomes necessary and it no longer is possible to be lost. So it may be
that's
something like that too.
This is this is not just answer. This is the answer.
Right.
Basically, you incorporate an amino acid because it's important for something. You don't incorporate amino acid just because you want to incorporate something. It happened to be important for some sort of function and makes perfect sense. As I said, you have a evolution of proteins starting from small peptide motifs, domains, and finally you have proteins. This evolution must be, of course, kind of follow during this research.
Right. Very interesting. Well, I no longer have any questions. I think we are almost past 1.5 hours. I don't know if. Yeah. I don't know if speak. Yeah. I don't know if speakers want to continue. Yeah.
Yep. We are at an hour and a half. Should we end it, guys?
I no longer have any questions, so I'm fine. Okay. Maybe somebody else has any questions or comments.
Yep. I have a million questions, but I have to. I have to draw them out, and I am too tired right now. So anyway.
Okay, guys.
Okay, I'm gonna end it.
Okay.
Goodbye, cruel world, right?
Goodbye, cruel world. Yep.
Stay curious.
Okay, see you guys.
See ya.